I'm here from the University of Windsor, which is 1 hour away from here. We're just across the border in Canada. So if anyone wants to come visit the lab sometime, you let me know. It's much closer than you think. I really enjoyed it and I learned a lot. But I became very critical of the methods that we were deploying. The work that we made during this time gave the same feeling as those Frankenstein movies that I had seen previous. So I wanted to think about other ways of making work that really maybe more truly reflect the complex bioethics, the biotechnological sphere, in a way that is not intended as fear-mongering, but in a way that is inclusive and asks us as a general public to participate and to think carefully about our role in that space. So I also wrote a dissertation on this work as part of my PhD, and I spent a lot of time analyzing the types of metaphors that we have for representing science to ourselves as very clean, cool, sterile, digital, perfect, virtual. But my experience of being in the lab was very different than that. That it was quite often hot and bloody and unruly, it was complicated, it smelled bad. Lunch got mixed in with the research projects. And so I wanted to start thinking about ways that I could create representations that could travel through the public sphere that might give a more general audience some of these other visions of biotechnology. The other thing I got thinking about when I was in that space was how maybe the lab was more of an ecology rather than, like, we think of labs as these perfect sterile places that are separate from the rest of the world. But actually, like, there's always like stuff coming in on the bottom of the scientist's shoe, or gum, or something that spilt a couple of years ago in the back of the fridge and nobody wants to touch. And I got thinking about how scientists could also be one of the organisms in that space, and that we are not necessarily the top of the food chain, that we're sort of interacting in like mutually beneficial and mutually detrimental ways with the other organisms in that space. So I started doing work that attempted to create that type of this alternative visions of the lab. And this is, I used to work at the University of Leiden in Holland. I was teaching a bioart class there. And in Holland, it's funny, they're really focused on animal rights. I couldn't do a lot of the cell-based research that I was doing in Australia. But the Dutch had no problem with nudity. So I took off my clothes every chance I got, because I thought it was really funny to think about how the lab coats and the outfits really present us as separate from the other organisms within that space. And I started doing these weird performances where I would do things in labs that you're not supposed to do, and I would do little performances for audiences that were not human. So these are trays of bacteria that I've been growing, and I invited them into the lab with me one night, and I did a little performance for them as my audience members. So this has kicked off a whole range of these sort of unruly performances in laboratory spaces. I often collaborate a lot. I collaborate with a lot of people. You'll notice all of these images sort of have different types of credits on them. This is with Kira O'Reilly. She is a British artist. She does a lot of performance and bioart, as well as object making. Kira and I met at Symbiotica, and we first learned how to work in a lab together. And we spent a lot of time crying because we felt really sad for the cells. So now, whenever one of us has a residency working in a lab, the other person will fly in and we'll do something weird in that lab space and do a lot of documentation. So this is when she was at the University of Birmingham in the United Kingdom in 2010. And here, they wouldn't let us take our clothes off, but we hired instead a dressmaker to create for us these sort of like kimono, origami lab coats. And we thought about all the different like sort of folding and unfolding of bodies and the folding and unfolding of proteins and going on within these spaces. So this is a molecular biology lab, and the two of us spent the afternoon wrestling in there with a really great photographer called Hugo Glendinning. And then these images, we liked them a lot, but we started to twin them, so they're actually digitally cut in half and then twinned later on in the studio. This is another day from that same photo shoot. This is in their animal tissue culture lab. And I think that, on the one hand, we're doing artworks in there for audiences and environments and galleries, But I think also a lot of the audiences are the scientists themselves. Like, can you imagine you're trying to get your work done on a Friday afternoon and I'm rolling around on the floor in the lab next to you? It quite often causes a bit of a kerfuffle, and we end up having really interesting discussions with the scientists. So to take those sort of like notions of a laboratory ecology further, I started thinking about how upsetting my role in the lab, but then maybe also taking organisms from the lab outside and giving them other sort of experiences in other ecological environments. So this is actually a little out of order. This is an installation that I did later on in that series where I started building labs in tents, and I would put them in different outdoor spaces. And this is actually for a gallery in Edmonton in Canada, where I built a lab in a tent in Edmonton, and I called up the local university, and I went down through the biology department directory, and I called each professor, and I asked them if they could donate some of their organisms from their lab. And so I made this sort of like Noah's Ark of all the different organisms that were working in the labs at the university at that time. And I took them on a little camping trip in the gallery. And then, you know, I took my clothes off and rolled around in the tent with them. So then I started doing a lot of these labs where I build them literally in the outdoors. So this is back in Leiden again in the Netherlands. While I was teaching the bioart class, we did a project called Inside Out Laboratory Ecologies, where we recreated mayonnaise luncheon on the grass, but we did it instead in a bioart flavor. And we set up these beautiful picnic tents, and we had classical musicians and, you know, flags and artwork dangling from the trees. And we invited children and regular people to come, and we had a free picnic. And then we also had 3 workshops where you could do DNA extractions from kiwi fruit, you could do plant tissue culture from tulips, and the last one was collecting bacteria samples from the local ecology. Again, trying to link bioart and thinking and participating in the biological sciences to other like long-standing practices in the fine arts, but maybe also into like non-art environments. Like a picnic is a good trick. It's a good way to trick people into coming to see some art and some science, because they're thinking, I'll get some free food. And the next thing they know, they're having a picnic and learning something about science. And then I started to get more extreme in terms of the ecological spaces that I wanted to take them to. I decided I wanted to find like nature, and I found that really hard to accomplish, because everywhere that I went that thought might be natural had actually already been colonized in so many ways. So this is Banff National Park in Canada. It's probably our most famous national park. This is one of the big vista views in that park. And I was thinking that really, you know, I was getting back to nature, and then when I started working in Banff, I realized that Banff National Park is actually a 100-year-old science experiment, and they've been controlling this corridor of land for a long period of time and conducting all sorts of experiments there. So really I found that I'd just gone into a bigger petri dish. But I wanted also like to connect these questions to the Canadian imagination. I think that Canadians quite often, you know, living next to their big brother, the United States, often feel like they can leave these larger technological and conceptual questions to the US to sort of sort out on our behalf. Because the population is so much bigger and there's so much more research money down here, I think Canadians don't see themselves as always connected to these questions directly. And so I started making a lot of work situated in Canada to sort of build more bioart in Canada, but also to have citizens from my country understand that they are also implicated in these questions. So I started doing these little funny pieces in Banff because I wanted to work with the Banff Centre for the Arts. And for those of you who don't know that, it's a mega art center, but it's in the middle of nowhere. And people come from all over the world to work there. And I wanted to convince them to host a really large project. And when you're working with a big institution like that, you kind of have to date them for a little while. So I do these little projects like that. And this one is called Cell Break. And it's where I did a tour with a bunch of people where we took lab specimens that I'd ordered in from 1-800 lab specimens. And we took them on a little day hike in the mountains. And all the specimens come in these little clear glass containers, and we let them see the view, we dipped them in the river, and we had a picnic with them. And then at the end, I asked each of the participants if they wanted to give their specimen a breath of fresh air. And those who did would open up the top of their little tube and let the air come in. And they couldn't dump it out, because that would be bad, but they put the lid back on. And of course, the fresh air to us seems like a really beautiful gesture to a lab organism. But to the lab organism, the fresh air is really contaminated, and it killed whatever was growing in the petri dish or in the test tube. So it sort of played with these ideas and notion of wildness and freedom, and you know, the types of harm that we can unwittingly project in these organisms. So after a number of years of doing little projects like that, I convinced them to let me do a big project. It was called BioArt Camp. And I brought with me 20 artists and scientists and students from all over the world. And we built a portable BioArt lab in the forest. And we camped up there for 2 weeks' time. And I really literally wanted to eat and sleep and live with our lab organisms. And I wanted those lab organisms to eat and sleep and live without, you know, the ecology and the bears and the bugs and the grass. And so I sort of selected these people through a variety of different processes where they all proposed projects. And so everyone there had their own projects that they were working on, and then we also together collaborated on some projects. We built this sort of hunting lodge tent in the forest. And again, I was really playing on the sort of colonial history of Banff National Park and linking that history to the types of actions that might be going on in laboratory spaces. For this work, but I have to say for every piece I've shown you and everything I've ever done, there is really a lot of paperwork in order to do BioArt legally into an institutional environment. I haven't shown you anything that's dangerous. And you can do all of these things in your backyard at home with your own time and your own money. But if you want government grants, and if you want to do them linked to a university and well paid a salary, you have to make sure that everything has been approved through all of the different levels. So for example, this one required permits from Parks Canada, not only to camp, but for us to collect things like rocks, and because you know Banff National Park is like a world wonder, right? It's like you can't just go and take rocks and leaves and do stuff with it. We need a biosafety assessment for the whole project. Everything from like, you know, we needed sprinklers in the tent so that if there was a fire in the lab, it wouldn't burn down and kill everybody. Ethics, each person's project had to be evaluated through human and animal research ethics protocols. So years and years of paperwork to make this happen, and really the types of things I ask large institutions to do, they never want to do them. So they always almost turn me down, and it's always a big fight. So this is the last piece of paperwork I received approved, you know, 2 days before the camp started that meant we could go on, but it was down to the 9th hour. So I want to show you that so that A, you know the stuff I do is safe, but B, I want you to know that when you ask that science and business and large government research institutions really like to sort of do what's been done before through codified means, right? And when you want to do things that have never done before, and you want to push the boundaries and break the rules, it quite often means years of convincing people and creating a paper trail that establishes your legitimacy in that space. So this is the camp in progress. You can see during the daytime, so we have our hunting lodge tent, and at night, inside of the tent, I've installed like very Alberta. Alberta is like Texas North, you know, so there's a lot of oil money there, and lots of big guns and that type of thing. And so that has that sort of feel to it, but it also has a fully functional laboratory in it, sterile work environment. We built this sterile hood ourselves and installed a picture, like a photographic panorama of Banff in the back of it. And then each of the artists and scientists went on to do their own project. This is Tammy Duff. She worked to collect local mycelium, which is like fungi, and she was interested in assessing different materials as possible biodegradable polymers that could be used to build lab equipment. Part of her argument in her work is that a lot of science is very disposable plastics, and that there's a lot of waste produced, and so she would like to produce plastics and or like disposables, but also large pieces of equipment that are made with biodegradable materials. Paul Vanus at the top here, he's an American artist from Buffalo, and he reverse engineered PCR, which is a protein chain reaction. It's a way of cloning DNA. You take DNA and you thermocycle it through these temperatures, like really hot and really cold. And what it does is it strips the DNA in half and allows the other halves to be filled up again, and you can clone DNA many, many times. It's in a very expensive machine that's patented. And he figured out and successfully built on the campfire that if you have 3 buckets of water and you keep them at the appropriate temperature, you can move your DNA through those buckets and it will work. And it costs about $20 more if you're drinking vodka at the same time. So there were all these types of projects, and some of them were really technical and scientific. Here we have Vilant Mutis in the middle there, who's a scientist who was creating these portable air quality testers to test for sulfur in the air. But then some of them were really evocative, like Ian and Louise Backstrom brought a wooden sculpture of a man and rolled naked with him in the forest and took photographs the whole time we were there. So a really wide range of projects. And then also I really designed a lot of the equipment. This is called an incubator in sheep's clothing, to try and reconnect the equipment and its functionality back to sort of the biological origins it's intended to reproduce. And then of course, you know, again, I really like to trick regular people into seeing art and science. So we had a free barbecue and campers came, like hundreds of campers came from all over the park. And we showed them all of our art science projects, and we had a songwriter who came and wrote science fiction, cowboy, country western songs about DNA extraction and whatnot, and they entertained us all night long. It was a great party. So the thing that happened at this camp that didn't occur to me, like I'm really focused on human interspecies relationships, right? And I forgot to account for the human-human relationships in this equation. And so when we got there, I knew I wanted to sleep with the algae, but it didn't occur to me that there was another organism at play, right? And that means us as individuals, but also our interrelations and maybe the social organism of the camp. And we became like this sort of like group think started to happen after we were out in the forest for long enough together. We started communicating through howling. There was a little boy who came with us, the son of one of the artists, and he liked to howl. And so we started howling, and then we found out that like if we went into the town site and it was time for everyone to leave on the bus, if you just stood in front of the bus and howled like a wolf, everyone would come running. So it started this sort of like, we got really smelly, and we were preparing food for each other, and then we were preparing agar in the same environment, and the boundaries got really blurred. And I got thinking about how that the care that I want to afford to these organisms also has to first be afforded to ourselves and to the other humans in that environment. And that maybe the type of interspecies care I would like to see afforded in a lab can't happen unless we also manage the human community within that laboratory environment. And so a lot of my work since the camp has been focused on some of those ideas. We have lots of sponsors. So we had Dutch filmmakers come, and they interviewed everyone and followed us all through our projects. And we made this really great 3-channel video installation that's been exhibited in a number of places. And everything has subtitles to it, but the sound has been orchestrated, so it sort of comes up and one person talks for a while, and then the other one will sort of overlap. And it becomes this very like multivalent, immersive experience for you to get that feeling of being in the camp with all of these things happening all around you. So while I was in the camp, my project, because everyone had a project, right? My project is I wanted to make a natural science collection of the camp. And I wanted each of these objects within the collection to create a taxonomy that spanned across different boundaries. So normally a collection, a natural science collection, would be like just of like local flora and fauna, let's say, of the campsite. So I did do that. But then I also collected all of the different biotechnological organisms that were growing in the lab. But then I also collected samples from all of the different artists, And some of those samples were biological, but some of them were cultural objects as well. Like, so for example, Bulant lost a filling while we were drinking around a fire one night, so he gave me that. Another one of the artists didn't shave the whole time we were there, and at the end he shaved his beard and he gave it to me. But then I also took things like, you know, apparatus that have been built by different artists to achieve different goals, or I took this funny toque from one of the artists who had worn this ridiculous toque the whole time we were there. And so this collection starts to make an argument to the audience. And the argument is that all of these things have some sort of equivalency, that they're not, one's not intrinsically better or more important than the other. So I started photo documenting each object and creating these sort of montages that would place these different orders of life all in the same environment. And then I started, you know, I did a variety of different types of montages. So this is, on the last day I went around and swabbed bacteria off of every surface in the lab, and then I had one of my assistants do some microscopy for me of everything. I started drawing all the different objects. And then we created a data system, like, so with a little sheet here where you have the photograph of the object, its, you know, weight and size and date collected and all that information, but also then sort of cultural stories about the object or biological data about the object all into one document. And I created this traveling natural history collection that would become a way to tell the story about the camp and to tell this story about all these orders of life to other audiences. And so here you can see is the video installation that I told you about, but within that same environment we have, so here is like a diorama of the camp, and it looks like a really big petri dish. So it's like Banff National Park in the petri dish. This is the bear that was in the back of the hunting lodge tent. I've attached a human anatomical spine with some of the specimens collected into it. And so it becomes this sort of like compilation of all of these sort of intersecting vectors into thinking about how life is interconnected in multiple ways. This is an owl nightlight from that collection. This is the conjoined jackrabbit microscope twins. So the microscopes are still, they're functional, and you can look at slides from different collections that were made at the camp. And then there's just thousands and thousands of all these little items that have all been categorized and numbered. So that's some of my own practice. And the other thing that I want to talk to you about today was the work I do at the University of Windsor. My teaching and my art are really entwined. And since I'd come to Windsor in 2008, I built a lab there. I got really tired. I was like living out of suitcases all the time. And I wanted to live near a lab, and I wanted to be able to do projects that took many years to grow. Because quite often when you do a residency, 6 months is as long as it can be. So I built a lab at the University of Windsor, and it's a studio and a lab that facilitates my research, but also graduate and undergraduate projects. We host a class out of there. It's called BioArt Contemporary Art in the Life Sciences. So it's an annual undergraduate class, probably similar to this, except for half of the classes are actually in the lab, and the students are expected to make their own BioArt projects at the end. We use the lab as an exhibition venue so that biological artworks that can't leave the lab can then be seen by the public. It's also a bit of an art installation. You'll see there's always like taxidermy and drawings on the walls and stuff like that in our lab. And then it's also a home base for all these sort of portable labs that I've been building. So this is the lab itself. It used to be a paper making studio, and now we have a ton of equipment in it. Some of it we built ourselves, and some of it is sort of like legitimate scientific equipment. These are my students. They're very enthusiastic. Windsor is a very interesting place. So like Detroit, Windsor is really blue collar. Its history is really blue collar, and it's very working class. And so a lot of my students are sometimes the first in their family to go to university. So sometimes like there may be, like when I used to teach in Montreal or in Holland, and a lot of the art students, they'd be like way cooler, and they knew a lot about contemporary art and everything. These students are not as cool and do not know as much about contemporary art, but they are so much cooler in other ways. because they grew up in this auto manufacturing environment. They're really makers. So Windsor, like Detroit, everyone sort of like builds their own vehicle on the side, or like people build their own barbecues, like really elaborate, weird barbecues in Windsor. You always see some guy going down the street with some bicycle he built himself that has stuff coming out the side of it. So these students all grew up in that environment where like their mom was showing them how to take vehicles